Welcome back to the class. We are discussing about the concept of plant tissue culture. As a continuation, in today's class we are going to start the concept of somatic embryogenesis. First of all, let us understand what is meant by embryogenesis. Embryogenesis refers to production of embryo. Genesis means production, to produce. So producing the embryo. Usually we get an embryo from the zygote. We get the zygote by the fusion of the two gametes, the male and the female gametes. So the male and the female gametes fuse to form the zygote, diploid zygote. The diploid zygote then develops into a diploid embryo. That is how the embryos are generated by nature. But here we are going to study about the concept of somatic embryogenesis. Somatic refers to body cells, right? There are two different types of cells. One is said to be the sex cells, another one is said to be the somatic cells. Sex cells are nothing but the gametes, the sperm and the egg, right? Or the male gamete and the female gamete. Whereas body cells are nothing but the somatic cells or autosomes. Here, what does this term somatic embryogenesis means? Producing the embryo from the body cells. That is what the definition goes like that, right? So while we discussed about the concept of PTC, the procedure of PTC, we have studied that any plant part can be used to develop an entire plantlet. We can take a part of leaf and we can culture it, make the callus, develop the embryo, then plantlets and an entire plant. So we are developing an entire plant from a small piece of leaf. The leaf is nothing but a body cell, a body part of the plant, right? So that plant leaf, the plant leaf, which is a body cell, has been induced to develop into an entire plant. See, if you speak practically, a new plant will be developed from the embryo which is formed by the fusion of the male and the female gamete. But here, through the PTC, we are developing the new plantlet from the body cells, right? The embryos are developed from the body cells or from the callus which is developed from the body cells, right? That is what we call it as somatic embryogenesis. And normally the embryo which is formed by this type of process, the somatic embryogenesis, which is not exactly similar to the embryo which is formed by the fusion of gametes. It will be slightly different, right? So let us see the definition. The development of embryo from the callus tissue grown from the somatic cells of the plant in laboratory condition is what we call it as somatic embryogenesis, right? So this is very simple. Here, the embryos are grown from the callus tissue, whereas the callus is developed from the body cells, right? The body cells are already diploid in nature, 2n in condition. The same way, if you develop the callus from the body cells, the callus cells are going to be diploid, and the new plantlet, what is going to be developed from the callus, is also going to be diploid, right? There is no crossing over, there is no recombination of the genes, there is no variation. That is why the plantlets which are produced by the PTC are exactly genetically similar to that of the parent plant. They can be even called as clones. Because there is in case until and unless we perform any other activities, there won't be any sort of genetical variation, right? So that is why we call this kind of embryogenesis which is developed from the body cells is said to be somatic embryogenesis. And the embryos which are formed through this technique is specially called as embryoids, right? So this can be noticed or this is performed in various plants. So in our book it has been given allium cepa, that is onion, maize, rice and different plants. They have carried out this experiment and the somatic embryos have been successfully generated and the new plantlets are produced from the body cells, right? And the new plantlets which are developed from by this process, somatic embryogenesis, is more or less similar, or more or less it is similar to that of the normal plant, right? There won't be any sort of major differences, right? It will behave, the potentiality will remain as such, right? So what are the applications of somatic embryogenesis, right? So first of all, it is easy to produce the potential plantlets within a very short period of time, right? Imagine if you want to grow a new plant, we have to sow the seed, right? And we have to collect the seed. For that, we have to wait for certain period of time. For example, if you take a mango tree, it takes 6 to 7 years to bloom and to give rise to fruits and seeds, right? Whereas by this technology, we can able to develop the plantlets within very less period of time, right? So that is the first and foremost benefit of this somatic embryogenesis, and moreover, it is also used to produce synthetic seeds, right? So that is what it is given in our book. So let us see what is meant by synthetic seeds, which is also called as artificial seed. Artificial seeds are the carriers of next generation, right? Seeds contain the embryo. The future generation of a plant is embedded inside the seed. The embryos of the plant are embedded inside the seed or protected inside the seed, right? So here we are developing the seeds artificially. So whatever the embryoids we have developed in the plant tissue culture have been protected with a covering, and it has been stored or preserved like a seed, right? So let us go into the concept. The somatic embryos, which is also called as embryoids, developed by the in vitro culture or encapsulated, right? So the young embryos which are developed by callus or which are developed by plant tissue culture or somatic embryogenesis are covered with some sort of chemical substances called sodium alginate or agarose gel or calcium alginate. These are the fluid-like structures, and when we immerse that small embryoids into that jelly structure, they will cover the entire embryo and looks like a small seed-like structure, right? So you can able to observe it in the image. So see, so all these are the artificial seeds. So you can able to see the young embryos which are covered by this sodium alginate gel. So it will resemble like a seed, right? And moreover, inside the chemical, that is the sodium alginate or chemical alginate or calcium alginate, the embryo will not get disintegrated or damaged. It will remain as such, and through the cryopreservation technique, it can be stored for a very long time, right? So that is why these artificial seeds are generated, and the artificial seeds are also called as synthetic seeds or synseeds, right? So this is one of the special technique which can be done to preserve large number of embryoids which are produced by callus. And what are the advantages of artificial seed? So speaking about the advantage, as I have already told, it is in a very short period of time we can able to generate more number of embryos at a minimum cost. And moreover, through certain modifications, we can also induce we can also produce genetically engineered plants, right? Usually they will be if you don't perform any sort of activities, the embryos will be exactly similar to that of the parent plant. But if you want to do, for example, if you perform the protoplast culture, you can able to develop a new plantlet, right? Hybrid varieties can be produced. So that is why it is mentioned here, easy way to produce genetically hybrid plants. And the genotype of the plant, right? So genotype refers to the genetic constitution of an individual, right? The plant genetic constitution, right? The gene content of the plant can be easily studied, right? Whether the parent and the offspring, what or the vegetative individual which is formed by the PTC, or both are exactly similar, the genotype can be studied in such a way, and easy to cryopreserve for a very long period of time. And after longer preservation, we can able to regenerate it without any problem, right? Whenever and wherever we need, and moreover, it is easy to transport from one area to another area. And the last one is to produce identical plants, right? Moreover, we can able to generate both identical plants can be produced, and if we perform any sort of genetical modification, even genetically modified plants can also be generated. And growth of the plants from synseeds are faster due to reduced dormancy, because each and every seed has a specific resting period, right? We can't say it as a complete resting period. It is some sort of a time period where the seed or the embryo prepare itself for germination, right? From the date of sowing and till it germinates, right? For example, if you sow some fenugreek seed, it will take within a day or two, it will begin to germinate. That 1 or 2 days or 24 to 28 hours or 36 hours is the dormancy period for that particular fenugreek seed. For example, if you sow the mango seed, it takes 4 or 5 days or even a week to get germinated. That period of 5 to 6 days is what we call it as a dormancy of a mango seed, right? Whereas such dormancy is not necessary for these kind of somatic embryos developed by somatic embryos covered with artificial seeds, because they are already ready for germination and it is holded by means of this chemical covering, right? Once it is kept under the new suitable medium, they start germinating and start regenerating the shoot and the root if it is supplied with the proper amount of phytohormones and other nutrients, right? So these are the benefits of artificial seeds, right? So in today's class we have discussed about the two concepts. One is about the artificial seed, and another one is about the somatic embryogenesis. Hope this will be a useful session. Let us meet in the next episode. Thank you.